So I would like to introduce Dr. Rezwan Tariq, who's a postdoc in the lab, and he's going to talk to you about the work he's doing to get everything ready for the GWAS that you've heard often here, which is basically to look at a very large number of rose genotypes, the genotypic data, and then, of course, combine it with phenotypic data to try to find, I guess, factors for resistance to rose rosette disease, black spot, cercospora leaf spot, etc., etc. And what he's going to do today is give you an insight on, not an insight, but show you some preliminary work he's been doing with black spot disease and some of the work that requires to go before you actually do that. Go ahead, Rezwan.
Good morning, all. Can you hear me? Yeah. So my name is Rezwan Tariq. I'm a postdoc in Dr. Oscar's lab. So today I'm going to present my work, like genome-wide association mapping work. So just briefly describe about my background. The cultivated roses actually has the value of the $28 billion globally. In 2014, it has the worth of $203 million, but it decreased to 17% in 5 years. In 2019, one decreased to $168 million. So what are the key challenges actually we have been facing right now? Is a black spot disease and the rose. The black spot disease actually mostly widespread foliar disease globally, especially in the hot and humid region, and it is caused by a fungal pathogen. Its symptoms are the dark spots, which is often followed by the chlorosis, and ultimately, when the disease becomes more severe, it actually causes the death of the plant. And about its resistance, we find some kind of vertical resistance gene on the chromosome number 1, 6, 5, and 7 region. And the partial QTLs were identified with the multiple linkage groups on linkage group 1, 2, 3, 4, 5, and 7 in across the diploid and the tetraploid rose population. For the rosette disease, already they have discussed a lot about the RRD disease. So I just skip the intro and move on. It is actually caused by the rosette amenovirus. Its vector is terrified mite and is distributed in the central and eastern United States region. And symptoms are the reddish, polypic, and twisted growth, shortened internodes at an increased number of the prickles here, and ultimately, which is flattened the stem structure whole. And about its resistance, a few criteria were found out which actually reduce the susceptibility of the RRD disease identified on the linkage group 1, 3, 5, 6, and 7, both in tetraploid and diploid population. So it's a main part, like here is our story, like what are the objectives of a study? We have to screen the rose germplasm panel for resistance to the BSD and RRD, and we use the ploidy-aware SNPs like triploid, diploid, and tetraploid genotypes for the genome-wide association studies, and we have to identify the genetic factors which are actually behind the RRD and BSD resistance. And based on this SNPs, what SNPs are actually present in the which gene actually. So we have to identify that them, and based on our whole, we have to make a prediction training data set. So regarding the phenotype, for the RRD, the population is being grown in the Oklahoma, in Cross Valley, Tennessee. Regarding the phenotypic data of the BSD, we have the huge germplasm of the roses, which included the breeding line species, varieties. Actually, we collect the data of the BSD from April to November in 5 years data. So we give the scaling from 0 to 9 of the infected foliage and give the 10% increment depends on the progression of the disease. So after collecting the data, phenotypic data, we estimate their blues from the SRML. And here is our distribution of our phenotypic disease severity score in the 5 years of the 2016, 17, 18, 24, and 25. It's showing this nice distribution of our data in 5 years that is used for our GWAS analysis. And regarding our markers data set, we have the 767 different genotypes, which actually representing the pool of breeding lines, cultivar, and different species. And we determine their polarity with a Q polarity. And from this, we ultimately find out their dosage calling from the fit poly. So we have the 68,893 total markers of the exome array data set. So based on the stats of our data, we keep the markers which has the less than 50% missing data, frequency, minor allele frequency is more than 0.05 or 5%, and the polymorphism information content more than 0.25, more than the scale. So based on this filtering, we have the 15,121 good quality markers for our diversity panel, which is around total 21.9% markers out of 68K. So this is our markers with that we will use as an input for our diversity analysis. So for polarity determination, as I mentioned, we determine the polarity of our genotypes. We have 161 genotypes are the diploid one based on the Q polarity, and triploid are the 74. And we have the more representative germplasm belongs to the tetraploid, which are the 532 genotypes belongs to them. So after determining their polarity, we did the diversity analysis. We find out the genotypic data to compute the genetic distance. Sorry.
So you can clearly see.
We can see it's a huge. It's huge.
It's huge.
Yeah. So because 767 sessions, so so we calculate their genetic distance, and from this genetic distance, we ultimately find out their relationship between the genotypes. And the principal component analysis is showing the clustering of the genotypes based on their PCA coordinates. We find that tetraploids are making a separate ball-like cluster than the diploid ones, and many diploids are separate clustered from the tetraploid ones, and we can see many diploids are actually mixed with the tetraploid ones. It may, because tetraploid we have in the roses, they are autotetraploids. So they are could be genetically same actually. So, but interesting part is the triploid ones, they are in the middle of the tetraploid and the diploid. And we can assume, or we can see they are the recent hybrids between the cross of tetraploid and the diploid ones. So from our genetic distance data, we can see a lot of tetraploids, they are making a separate cluster, quite different from their, and they're totally divergent from the diploids ones. And but we can see here, few triploid ones are in the tetraploid cluster, because when they make a hybrid between the diploid and tetraploid ones, most of the genetic proportion is coming from the tetraploids. And here, if we see about the diploid cluster, a lot of the diploids, they are quite divergent, are different genotypically from their diploid other cluster, which make actually a good story about it. And the many triploid ones, they are quite making a mixed cluster with the diploid, lot of and few tetraploids in the in this phylogenetic tree. So it's showing the whole story about how they are genetically same or genetically different to each other, or divergent. So here's the kinship relationship between our genotypes. There are basically two big main clusters. One is with the tetraploids, few chunks of the triploids in it, but it's mostly representative of the tetraploids. And the second cluster is actually occupied by the diploids and the triploids. Here, actually, we can see a few subset of the scratches that are showing a kind of, they are the hybrids are the ancestry proportion of the admixture between the tetraploid and the diploid ones here. And even we can see some scratches of the high relatedness here. But as we can see, there is a high relatedness within the cluster, then between the cluster. The thing is this, there is a limited polarity gene flow between the cluster. So the genetic structure of the based on the polarity of the tetraploid, triploid, and diploid ones, we actually identify the K membership based on the SNMF model of the cross entropy of the K membership. We use the first K6, 2, 3, 4. By mathematically, we can see we use the elbow method. Highly significant, the difference cross entropy 3, 4, 5, 6. I use 6 one to actually remove the biasness from our sub membership of the K method, K membership of the structure data. As we can see in the K2, there are two membership, but the triplets were in the between them, and it's quite clear in the K3 structure that the tetraploids, which are actually showing the proportion of the true diploid tetraploid ones here, the blue color we can see, and the yellow part and the part, most of the part from the diploids one here, it's showing they are the hybrids between the tetraploids and the diploid ones. But in K4, K5, and K6, we can see that the tetraploids, they are making a good representative clustering, and the population is almost stable between the cluster in the tetraploids. Whereas in the diploids, we can find some kind of integration in the diploids, but they making a quite clear clustering. But if we see in the K4, 5, 6 in the triploids, they're making a transitional sharp peaks. It's showing that these are the specific output of the specific cross between the tetraploids and diploid ones, and this population of the triploid is not very stable. So here's our Jiva's work of the black spot resistance in multiple years. So the Venn diagram is showing the overlapping samples and the genotypes we have across the 5 years. We can see the lot of samples were taken in the 2025. They are quite unique, other compared to the 4 years past. So we have the data from the 16, 17, 18, 24, and 25. In 2016, we used, we had the data of 225 genotypes, diverse genotypes. And based on this, we find actually based on the p-value 0.05 probability, on our threshold, 3 SNPs were found. 2 SNPs were found on the chromosome number 1 and 1 SNP on the chromosome number 6. And in 2017 data, we applied 276 genotypes, and based on our results, we find that 4 SNPs on chromosome 2 on the chromosome number 1 and 2 SNPs on the chromosome number 2. So for the data of 2018, we find only 1 SNP on chromosome number 1. That we can find another SNP here, which is the same one, because we run 2 models, additive model and dominant based model, to find out the significant SNPs. They are the same, but the 2 models we run to get as much as we can, the maximum SNPs. In the because we didn't find any significant SNP in 224, we had the 196 accessions in 2024. So in 2025 data, we had a lot of 484 genotypes, and we find only one SNP on chromosome number 2. But for across the year blues, like 5 years data, we find the two SNPs, significant SNPs on the chromosome number 2 and chromosome number 6. So after that, our next target was to find out which gene are actually corresponding for these SNPs, where in which gene these SNPs are actually situated. So we find the predicted SNPs actually 11 SNPs, but I mentioned that 10 SNPs here because 2 SNPs are actually repeating in 2 years. So 10 SNPs are here, their putative genes and what the putative description or function of the annotation of these genes I mentioned here. They have the quite different function in across the genome. So based on our predicted co-expression, how the genes actually interlink to each other. So I mentioned this gene names with the SNP so that the audience can understand which SNP is actually used here and which gene is actually making us a good network with the heat shock proteins, which actually play a role under the pathogen stress, pathogen infection. And the second SNP we find they have the good network, a strong network with the ROS responsive genes, which has a good role in the stress signaling. And this SNP responsive gene, they are making a good network with the DNA-directed RNA polymerase, nucleotide binding proteins, which play a role in the RNA processing or transcription of the RNA. So our 4 SNP is actually making a good cluster with the clathrin protein, beta-galactosidase, which these are the which genes actually play a role in the cell wall remodeling. And our 5th one SNP has a good strong network with the calcium trigger signaling. And we can see in the calcium trigger signaling, they have when the pathogen recognition receptors actually receive the signals from the pathogen, they attack, and the calcium influx become very high, which ultimately trigger the calcium-dependent protein kinases and other kinase responsive gene, which actually cumulatively role to enhance the resistance mechanism in the plant. And our other SNP responsive gene, they are making a network with the genes which have the role in the stress response signaling to play a role in the membrane remodeling. The third last one has the role in the making a network with the glycerol 3-phosphate, xylotransferase, and these kind of enzyme responsive genes. They have the role in the plant metabolism mechanism. And second last gene we find they are playing role in the ribosome making subunits, which have the ribosome has a role to have the protein synthesis process. And our last one has the strong network with the cellulose synthase responsive genes, which have the quite good, which have the role in the cell wall remodeling. So finally, here's a summary of our phylogenetic analysis, just the differentiation between the tetraploid and diploid ones, and our genetic structure work suggested that the germplasm panel is subdivided into the various subpopulations. And we find with the Jivas, we find 10 SNPs, 10 unique SNPs, chromosome number 1, 2, and 6, which are associated to the resistance to the BFP. And 4 SNPs actually quite play a role in the plant innate immunity, and other 6, they are involved to support the plant immunity, like the cell wall remodeling related. So this framework we will use for the RRD resistance work as well, because we have the data, and next our plan is to analyze the RRD data. So I'm very thankful to our team like Yvonne, Natalie, Pamela, and Jennifer to make nice picture.
We know what you like.
Well, not really, you were a student worker like, what, 3 years ago?
Mhm.
Yeah.
On the vault.
That's a-
So they maintain a nice field, you will see in the field, and they collect the data, help out for me the collection of the phenotypic data as well. And I'm very thankful to Jeetal Law. I just met one time him physically in the lab. So he's working in the USDA right now. Very thankful. So it's just start of my work. So let's see, we have more finding in future. And I'm very thankful to our collaborators and funders who provide the great funds to actually to do this kind of nice work. So hopeful we have nice, good work and findings in future. Thanks a lot for having a great patience.
We do have some time for a question, but at the same time, given that we're caught up on time, I think we could probably take a 14-minute break and come back at 10:50 for the last talk of the day. Go ahead.
Hi, Dr. Rezwan. Nice presentation.
Thank you.
So you have several years of data. And how many of those genes are stable? I mean, how many of those genes were clearly identified amongst the years, amongst the locations?
So we have the data of the colonization data of the one location. So we have only the data of the 5 years of the colonization right now. For the stability, we have to check for the multiple location. But right now, we just analyze with the colonization data.
Okay, so location was what? So year was different. So are any of those SNPs common among the year?
Yes, one SNP, as I mentioned, SNP 718 is actually in 2 years, is quite stable and find the significant one.
So my another question is, how did you determine the model that you are using to predict the association? Because some of the models could lead different SNPs as well. So have you tried different models to find out if a SNP that might be a commons, like more commons amongst the year? Because if you have like 5 or 6 years and find a SNP only common in 2 years.
Yeah.
Okay. So regarding the SNP model, we use the good model like you mentioned the GWAS poly, additive model, and dominant one. Dominant is alternative dominant and the perfect dominant model. So these are the 3 different. So based on the 3 different, we find that the ones which is quite stable in 2 years. But when we get the multiple locations data, then we can predict, yeah, these our SNPs are quite stable across the years and across the location in the US. So this is just the preliminary results we have right now.